Hello folks, and welcome back to another Learn with Lou video. Today we're going to be going over the 2021 Section 1 for National 5 Biology. Before we get stuck into it, remember that you can pause the video and go through these questions yourself, and then you can check with me and the correct answers. Where possible, I will try and work through the answers, showing any math involved, etc. I would also encourage you to always just have a go at these questions again and again and again. There's nothing better for your revision than repeating the past papers, and especially the ones that you really struggled with. In the video description, as usual, I will link to the question, so if you want to skip straight to that question, then feel free. But other than that, let's get into it. So section 1, question 1. Which row of the table describes the effects on red blood cells if they were left in the liquids shown for 20 minutes? So again, you can pause the video here if you'd like. Okay, so if you pause the video and have a little go, let's see how you get on. So, with multiple choice, I always like to cancel out the correct answers. Sorry, cancel out the wrong answers. Don't cancel out the wrong ones. So cancel out the wrong answers. We want to be going for A, B, C, D, multiple choice, nice and easy. So we have got a red blood cell. Okay, so it's very important that we've got the red blood cell. So that tells us it's going to be an animal. And now animal cells don't have the cell wall, so when we put them into pure water, they're going to swell and they're going to burst. So we know that the answer for the pure water section is either going to be A or B, and we've already gated out C and D. If we put it into the 10% salt solution, well, only plant cells can plasmolyze in this case. So we're going to be looking for the shrink. So that's going to be answer B. Okay. Question number 2. Which of the following statements is true for a DNA molecule? So again, pause the video, see how you get on. Okay, now that you've paused the video and had a little go, let's see how we go on. So, which of the following statements is true for a DNA molecule? So we've got A, B, C, D again. We're going to try and cancel out the wrong answers. So A says it's a double-stranded and found in the nucleus. Well, that's actually, funnily enough, the correct answer. Okay, but let's just check over the rest of the answers just to double check. So it is a single-stranded and found in the nucleus. Well, DNA is double-stranded, so we know straight away that that can't be correct. It's double-stranded, correct, and codes for making sugars, incorrect. So the first part of that statement is correct, but the second part is incorrect. It is single-stranded. Again, we've already had that conversation when it came to answer B, so we know that the answer must be A. Question number 3. A molecule of DNA has 4,450 bases, of which 890 are cytosine. The number of adenine bases in this DNA molecule is, and again, you can pause the video and see how you get on. Okay, now that you've paused the video, you probably broke out your calculator for this one. Remember, it's always important that you have a calculator to hand for the exam and for doing the past papers. But we've got 4,450 bases total. Okay, we know 890 of them are going to be cytosine. So 890 equals cytosine. Okay, but C always goes with G, so guanine. So we must have 890 of them. So we'll do 890 multiplied by 2. That gives us 1780 total bases. So we want to do 4450 minus 1780. So I'll just quickly put that in my calculator here. Helps if I know how to work the calculator. 4450 minus 1780 equals 2670. Okay, so that's the number of bases we have remaining after we've looked at C and G completely. Now we have to split that in half because the question has asked us about adenine. Well, what we've got here is adenine and thymine. So we need to split that in half. So we just simply take that number 2670, divide it in half, which leaves us with a grand total of 1335. Okay, so that answer leaves us with B. Alright, if you're not sure about that one, please message in the comments and I can always make a little separate video on that. Question number 4. Which of the following statements matches a type of protein to its function. Again, pause the video, see how you get on. Okay, now that you've paused it and had a little go, let's see how you get on. So with question number 4 here, which of the following statements match the type of protein to its function? Receptors target specific enzymes. Well, an enzyme is a protein, but the receptors do not target enzymes. Okay, and receptors aren't really involved in enzymes, especially at National 5. So we're going to be looking that A would be incorrect. Enzymes carry chemical messages in the blood. Incorrect. Enzymes speed up chemical reactions. So B has to be incorrect. Antibodies destroy specific pathogens. Now that one is the correct answer. So antibodies destroy pathogens. So an antibody is a type of protein that your body produces in its immune system, which fights against pathogens. Let's just double check though. So hormones carry electrical impulses. No. So hormones carry chemical messages in the blood. And electrical impulses relates to the nervous system. So C is the correct answer for question 4. Question number 5. A student set up an experiment to investigate the breakdown of starch by the enzyme amylase. So we've got this experimental setup here. And it's asked you which row in the table describes the contents of a control tube for this experiment. Okay, so pause the video, see how we get on. Okay, so if you pause the video and have a little go, let's see how we get on. So a student set up an experiment to investigate the breakdown of starch by the enzyme amylase. So we've got A, B, C, and D again. So just to point that out here. And in the top of the table is to give you what solution we're talking about here. So we've got a starch solution, an amylase solution, or we've got the water. So we want to have a look at our initial one. We've got 10 mils of starch solution. We've got 1 mil of amylase solution. Now, a good control of this, so we want to think about the experiment first off, we think of control. So it's the breakdown of starch by the enzyme amylase. So which experimental setup is going to prove that it is amylase that is actually doing the breaking down of starch? So we still need our starch. So we're going to need either B or D. So they're potential answers, which scribbles out A and C initially. Now, our amylase solution, there's no point having the amylase solution because if you take away the amylase solution, you're then going to be able to prove that it was the amylase that was breaking down starch. Because if you take away the amylase solution, there won't be the breakdown happening, so starch will still be present. You can still test for it, but in question C, answer C, they have left the amylase in there. They've actually left 10 mils of amylase, so they've added much more of it compared to the starch solution. So what they've done is they basically swapped them around, and you're not really going to prove anything at that point, and you're not controlling anything or showing me that this experiment is in any way valid. So what you want to be doing is looking at removing the amylase solution. So our answer here is B. And also note, when it comes to these sort of experiments, that we want to be keeping the volumes the same. So in this column here, they've added a water column. Now you notice in the original experiment there is no water, but you have to keep volumes the same. Now water is not going to react with starch, so we just add 1 mil of water to replace that 1 mil that was taken away from the amylase, and that keeps all volumes the same, and it allows for the validity of the experiment. Number 6. So the diagram shows part of the process of genetic engineering. So we've got the chromosomes, the source chromosome, and the required gene extracted from the source chromosome. The extraction of the required gene from the source chromosome involves the use of, and you can pause it right here to see how you get on. Okay, now that you've had a little pause of it, we're going to have a look through the answers. So the extraction of the required gene from source chromosome involves the use of bacteria. Well, bacteria are going to be involved in genetic engineering, but they're not going to be specifically involved in the extraction of the required gene from the source chromosome. You know, that's not even going to be involved in that part of it. You know, bacteria won't even be involved in that part. So bacteria is a no on that one. Hormones, nope, not involved there. That's just a nice random answer that the SQA like to throw at you. Plasmids is one that you might trip up with here because you talk about genetic engineering, and now plasmids is a big word. It gets thrown around with genetic engineering, but plasmids require the bacteria. Now we already know that there's no bacteria going to be in this experiment here, because we're talking about the source chromosome and taking the gene from it. Now, the gene is not going to come from the bacteria. It's going to come from, let's just say we're talking about insulin. That's the usual example given. It's going to come from potentially a human. We're going to take that source chromosome, that gene out of the source chromosome, and it's not going to have a plasmid because humans don't have plasmids. So plasmids is right, which leaves enzymes, which is the correct answer. Okay, number 7. The diagrams show 4 experiments carried out as part of an investigation into the rate of respiration in yeast. So you've got 4 experimental setups there. My suggestion to you right now is to actually just pause the video and have a look over these experimental setups and the answers and see what you can come up with. This might take you a little bit longer, but if you are quick about going through the multiple choice, you'll build up a lot of time. So when you're stumped with questions like this, it do take a few minutes to kind of look through. You generally will be okay. So again, pause the video and see how you get on. Okay, now you pause the video and have a little look. Let's see what you got on. So, which 2 experiments should be compared to show the effect of temperature on the rate of reaction? So we're looking for temperature on the rate of reaction. So the only thing we want to see change here is going to be the temperature. So if we have a look, we're going to have a look at 1 and 2. With these sort of questions, I always go through again all A, B, C, and D and just double check. So look at experiment 1 and 2. So experiment 1, experiment 2. So we're looking water bath at 22, water bath at 22. Well, you're not proving that temperature is going to be any different now. The only thing they've really changed there is the percentage of the glucose solution. So we've got a 5 and we've got a 2. That's not going to be any use to us because we're looking for the effect of temperature. We want to change the temperature. So A is out. So let's have a look at answer B. So we've got 1 and 3. So again, we've got a 22 degrees Celsius experiment 1. And we've got 30 degrees. Oh yes, fantastic. We've got a difference in temperature. But unfortunately, we've actually got a difference in the glucose solution also with a 5% in experiment 3. When it comes to this sort of experiment, we want to always make sure that we're only changing one variable when we're trying to compare here. So if I try to, for example, yes, I've changed the temperature, but how do I know it was only the temperature that affected the experiment? How do I know it wasn't the glucose solution that I changed also? So we only want to be changing the temperature. Right, so that leaves the B is out. So let's have a look at 2 and 3. So we've got a water bath at 22 degrees for experiment 2. And we've got 30 degrees again, so fantastic. We've got that. We've got 1 gram yeast with 5% glucose solution. Fantastic. We've got 1 gram yeast with 5% glucose solution. So C is looking pretty good because it is only temperature that has changed. So I'm going to put a little dot next to it. And let's just check over D. We've got 1 and 4. So 1 we know was 22 degrees. 4 is 30 degrees, fantastic. But again, we're seeing that change in the glucose solution. So we don't, and sorry, not even in just the glucose solution. Sorry, we're not seeing the change in the glucose solution. We're seeing the change in the yeast and how much yeast was put in. So we're seeing 2 grams of yeast and 1 gram of yeast in experiment 1. So again, like I said, it's hard to prove that it was just your temperature that was affecting the reaction rate there. So C is the correct answer. Moving on to number 8. The graph shows the number of bacteria growing in a fermenter over 30 hours. For how many hours was the number of bacteria 4,000 per millimeter cubed or higher? Okay, so you can pause the video here. If you need to use a calculator, feel free, and we'll see how you get on. Okay, so we want to have a little look at this. So we want it above the 4,000. So 4,000 is our key one. Now, don't be afraid to highlight and underline questions as you go here. So for example, if I was an exam, I might take my highlighter. Just go like that's a really important number there. Okay, so it has to be at 4,000 or above. So here's our 4. So the point where it goes above 4 is here, and the point where it dips below 4 is over here. So I'm looking for my time difference between, so like this line here, so bring it down there, and this line here. So we've got 15 to 20 to 25 to 30. So we've got 5, 10, 15 hours so far. And now each block is worth 30 minutes. So that means that is going to be 1 hour there. So we've got 15 plus 1, which equals 16. So what answer comes out of 16 is B. That is the correct answer right there. Notice how, yes, it's easy for me on an iPad to zoom in, but obviously you will have your paper in front of you. You can kind of look at it a little closer. But please do take your time when it comes to graph reading. The SQA are very good at putting in very tricky questions, so take your time. Okay, question number 9. The following key can be used to identify bacteria. So again, what I'm going to do is I'm going to zoom out a little bit here, and then you can see the whole picture. And what I want you to do is just work through that key by yourself, pause the video, have a little work through, see what answer you come up with, and then we'll work through it together and see how we get on. Okay, now that you've paused the video and had a little go, let's have a little go of it ourselves. So, the following key we use to identify bacteria. So we've got this individual cell here. Now, we always read the first question and work through. So, individual cells are spherical in shape, or individual cells are rod-shaped. Well, we can see from this diagram that they are spherical in shape. So we want to go to question number 2 now. Cells are arranged in a single chain, or cells are not arranged in a single chain. Well, obviously, this is not a chain. A chain would be more like this. Okay, so we want to go to question number 4. So we skip number 3 completely. Cells arranged in pairs, or cells arranged in groups. Well, it's very much in groups, yeah. It's not in pairs, so we're not seeing them just pair up like this. We're seeing very much them clumped together in groups. So I'm going to say that that's staph. And I'll just check here. So we've got 9, which should be B, which is correct. Okay. Number 10. A body cell of an organism containing 24 chromosomes is represented as 24, which is being encircled. So again, if I was in an exam, I would just be putting a little bit of highlighter on there. Saying, right, that's really important. I need to pay attention for that. Which column in the table represents the number of cells produced and the number of chromosomes in each cell after 2 cycles of cell division? Okay, so again, you can pause the video, have a little look, see how you get on. Okay, now that you pause the video, how to go, hopefully you're going to get the correct answer with me. So we're going to go through, so we've got A, B, C, or D to work through. So we've got the original body cell in the first bit, we've got after 1 cycle and after 2 cycles. So the original one, we already know starts with 24 because we highlighted that above. So we know that every single answer in the original body cell is correct. So we need to look at the next step to see what we can come up with. Now there is actually only one correct answer here. And if you get it, it's going to be this one over here for D. Okay, we've got 1 cell and it splits into 2 cells. Okay, you got 2 daughter cells. The chromosome number must stay the same. So the diploid chromosome complement must stay the same. Otherwise, you might change organism and things aren't going to work out very well for you if you're dividing and changing your chromosome number. So the only correct answer is D, because even in the next step, the chromosome numbers stay the same. Whereas you see in, for example, answer C, they've split it in half and then they split it in half again. Now that's incorrect. Okay. So D is the correct answer for this one. Number 11. Okay, so number 11, which row in the table shows the effect a change in blood glucose levels has on glucagon production? Again, feel free to pause the video just here and see how you get on. Okay, so this question actually has 2 correct answers. Yeah, you heard me right. This is a multiple choice that was a little bit strange when it came out in the paper format. It had already been processed and they did make a little bit of a mistake here. So if you ever look at the marking instructions, you'll see that question 11 has an asterisk next to it. And if you look at the bottom of that page, it does say that the answer, there's 2 options here. So I'm going to run through it. So there's 2 answers. I'm going to tell you straight away it's going to be A or C. Okay, so B and D are incorrect. Now let me explain. We've got a change in blood glucose level. So if your blood glucose or your blood sugar decreases, your glucagon production is going to increase. Okay, glucagon is going to break down that stored sugar that you had earlier. Okay, that it's going to break it back down to glucose. So it's going to break down the glycogen back down into glucose. So that would be a correct answer. But the opposite answer actually appears in this question. So if we have an increase in blood sugar level, well, that glucagon is also going to decrease. So we're going to have the opposite effect going on there. Hopefully this never happens again. But yeah, so there was a little bit of muck up there. And in all fairness to the SQA, they did accept both answers. So if you put A or C, then you were correct. But you had to pick A or C. You couldn't pick both. Okay. So number 12. Which graph shows the effect of increasing temperature on the rate of respiration in germinating peas? Feel free to pause the video again and have a look over. Okay, now that you've had a look over it, let's see how we get on. So, as we've got the rate of respiration, so we're looking at rate, so we're looking at, how fast is it going, what's happening, and germinating peas. Okay, so remember, we're also looking at increasing temperature. Now, temperature is a bit of a funny when we look at respiration because something bad can happen. Okay, so if I look at A, my rate of respiration is increasing at the same time as the temperature increases, and it just continues off into that abyss. It's not actually how that happens, because all you're saying there is if you warm yourself up, your rate of respiration will continue to increase. There will be an end point. And B is the inverse of this, so we've got the rate of respiration decreases as temperature increases. That means the colder you get, the warmer you get, the less respiration you're producing. So that's not actually happening. C is a little bit of a weird one. So it says that as respiration decreases until a certain temperature, then increases again. No, that's completely incorrect. But D is the correct answer that we're looking for here. So this one just here is what we're looking for, because what's happening here, my rate of respiration does increase with temperature, but it will get to that certain point where it becomes too hot, and then what's going to happen is the enzymes that help with respiration are going to denature, and then you'll see this sudden drop off of respiration beyond this point. Okay, so that means that the enzymes have denatured and the reaction can no longer occur. So D is the correct answer. Question 13. Which of the following is regulated by the medulla? Okay, so have a little think about it. Pause the video, see how we get on. Okay, so question 13. Which of the following is regulated by the medulla? So we've got A, B, C, and D. So thinking, breathing, hearing, and balancing. So thinking is not going to be a correct answer. That would be our cerebrum that is doing that. So that's like that conscious thought part of the brain. Breathing is actually our correct answer here. So it's the medulla that controls breathing. But as usual, we're just going to run through the other options just to be absolutely sure. So our hearing is actually something that we can that we do study at National 5. And D is balancing, so that would be our cerebellum, which is that rear part of the brain that we see in the diagrams. So A is incorrect, C is incorrect, D is incorrect, and B would be our correct answer for this one. Which features are found in phloem tissue? Okay, so again, pause the video and see how you get on. Okay, now you pause the video and see how you get on. Let's go through it. So, in phloem tissue, we would be looking for sieve plates and companion cells. So I'm going to put a dot next to that, and we'll just see how we get on with the rest of the answers, just to make sure that we are 100% with A. Hollow tubes and companion cells, nope. So we would be looking for the hollow tubes, nope, that's not correct. Sieve plates and lignin, nope, not correct. And hollow tubes and lignin, nope, not correct. So I'm more than confident that A is the correct answer at this point. The diagram shows the main parts of a flower. So we've got gamete J being produced in that particular labeled part, which row in the table identifies gamete J and its site of production. So again, pause the video and see how you get on. Okay, so gamete J is in the female part of the plant. So we want to be looking at not pollen. So we know it's going to be an ovule. Okay, so we know that gamete J is an ovule. And it's in the female part, so it's not the anther. So this part here is the anther. This part here is the ovary. So we know the answer D must be correct here for question 15. Let's have a look at question 16. Maize plants can vary in height. They produce either purple or yellow grains, and grain shape can either be wrinkled or smooth. Which row in the table identifies the type of variation shown by each of these characteristics? Again, pause the video, work yourself through the question, and see how you get on. Okay, so we're looking for height, grain colour, and grain shape. Now, the way I kind of think about this when it comes to height is I was never born over 6 foot tall. I was continuously growing. So that means that height must be continuous. So we're going to say that it could be either A or C, because it can't be B and it can't be D. The grain colour, generally, you don't change colour. Okay, generally grains won't change colour. So we're going to be saying that that's going to be a discrete. It's always going to have been that colour. So answer C is looking pretty good. I'll put a little dot next to that. And grain shape, again, it's going to be staying about the same shape. Okay, as it starts to grow and starts to develop, generally it will start to stay the same colour. So answer C would be the correct answer there. Okay. Question 17. Which of the following statements describes a feature of red blood cells? Okay, work yourself through the question, pause the video, see how you get on. Okay, now that you've paused the video and seen how you get on, let's have a look. So they are biconcave to absorb more oxygen. Ding ding, we have a correct answer. But again, let's just double check. They bind to oxygen to form hemoglobin. No, the red blood cell itself does not bind to the oxygen to form hemoglobin. And the hemoglobin is the actual part that binds to the oxygen to form oxyhemoglobin. So no, this answer is not correct. They carry out phagocytosis. Nope. Red blood cells do not carry out phagocytosis. They are part of the immune system. Nope. They are just part of your blood system. They are biconcave to absorb more oxygen. So it's 17. Right. Number 18. The diagram represents one alveolus in the lungs. Which two arrows show the direction of flow of carbon dioxide produced by respiration? So we've got A, B, C, and D to work through again. Pause the video, see how you get on. Okay, now that I've had a little look at question 18, let's have a look through it. So we've got 1 and 4. So which two arrows show the direction of flow of carbon dioxide produced by respiration? So 1 is saying that carbon dioxide comes in, and 4 is saying it's going into the blood. We know carbon dioxide is not going to go into the blood. We exhale carbon dioxide. So we're looking for it to go out. So there must be a 3 in there, and there must be a 2 in there. So we're having a look for 2 and 3. So that must be answer C. Number 19. The total variety of all living things on Earth is described as the what? A, B, C, or D? Have a pause, have a think, and have a go. Okay, now you've paused, have a think, and had a go. It is biodiversity. Okay, I'm not really going to go through the other ones because they all mean different things, but biodiversity means biological, and diversity means, variety, the different things that are going on in life. So biodiversity are basically two words mushed together. Number 20. In an investigation into density of planting, it was found that the lower mass of grain was harvested per plant when the density of wheat plants was at its highest. This was due to A, B, C, or D. Have a little go, see how you get on. Okay, so if we look at this question, it's got obviously 4 possible answers. So less disease. Well, we don't really know anything about what's going on with the disease there, so that's kind of a wild assumption, and I would probably find it very unlikely that the grain would, be really affected by disease in this sort of way. So A, I'm going to say no. B, more nutrients available. Well, we would see the opposite effect there. So if there was more nutrients available, we would see a larger mass when the density of wheat was at its highest. Less space for weeds. Well, no, we're not. We would actually see possibly the same result there. So no, we're not saying C, but more competition for light and nutrients. Now, obviously, competition is something that you do look at in National 5. We're looking at interspecific and the intraspecific competition. So in this case, we're seeing the intraspecific competition between the species of grain. So that's the wheat plant. So there's too much of it basically growing in this field, and they're all fighting. Well, not fighting, that's a terrible word to use. They're all competing for light and nutrients. Now, question number 21. The diagram shows part of a food web in a freshwater ecosystem. A disease caused most of the dragonfly larvae to die. Which of the following describes the most likely effect on the copepod and diving beetle populations? So we've got A, B, C, and D. Let's see you work through it again. Pause it. Okay, back at it with question 21 then. So if you've had a go, let's see how you get on. So if we are looking for which describes the most likely effect on copepod and diving beetle populations if the dragonfly larvae were to die. So we've got the dragonfly larvae going down. So what's going to happen to copepods? Well, copepods are consumed by dragonfly, so I'd kind of expect them to go up. And diving beetle, let's see if we can find it. So diving beetle consumes dragonfly larvae, I'd expect them to go down. So copepods would increase, diving beetle decrease. So we've got a potential answer for C, which is the correct answer. Okay, moving on to question number 22. Which of the following statements would not be suitable for advice? Not be suitable advice for surveying an area using a transect line. Okay. So pause the video, see how you get on. Okay, now you've paused it and had a go. Let's see how you get on there. So, A, throw the quadrats randomly onto the survey area. Okay. Well, we'll put a little dot next to that because we'll come back to that one in a moment. Let's have a little look over B. So carry out the survey more than once. Well, that's actually good advice. I would definitely give that. So no, I would say that would be an incorrect answer to give there. Use the same size quadrat at each sample site. Well, no, again, that's good advice. You don't want to be changing quadrats size. Take lots of samples along transect line. Again, that's really good advice to give. So, A would be the correct answer for this one because throwing the quadrats randomly at a survey area when you have a transect line. Now, a transect line is basically, so you've got point A, point B, and you've got a line. Usually like a piece of string, and it's kind of tied between two points of reference. And then you would walk along that line, taking paces. You know, you'd agree the size of pace you would take, and you might just place the quadrat down on that sort of transect line there. You wouldn't want to throw it randomly on a sample site. Maybe you throw it miles away from the transect line. What's the point in the transect line if you're just going to throw the quadrat randomly? Right. So question number 23. A marine food chain is shown. The quantity of energy available to the killer whale is the energy that the seal used for what? So A, B, C, or D. Work yourself through it. See how we get on. You can pause the video. Okay, now you pause the video and have a little go. Let's see how we get on there. So we've got plankton, cod, seal, and killer whale. So we're looking for the killer whale has eaten the seal. So what energy did the killer whale get? Well, the seal's already burned some energy swimming, so it's not going to get that back. The seal is trying to keep warm. Okay, so that heat energy has dissipated into its environment. You're not going to get that back. You are definitely going to get some growth energy there. Catching cod energy? Nope, absolutely not. It's going to be gone. So it is the growth energy there. Okay, so it's the only energy that the seal is used for growing and getting as big as it was to be eaten. The killer whale is going to be able to take in. Question number 24. Which row in the table identifies the effects of an algal bloom in a freshwater loch? So we've got A, B, C, and D, and have we work through it. Pause the video, see how you get on. Okay, so we've got A, B, C, and D for question number 24. So light levels, we've got algal bloom. So we're looking at light levels, number of bacteria, oxygen levels, and a variety of other species. So our light level for this is going to have to decrease. So we're going to get the algal bloom. So if we can imagine, we've got a loch here, okay, some water, and that algal bloom, I'll just change colour pen. That algal bloom is forming over the top and it's blocking out that sunlight coming in. Okay, so what's going to happen there then As all these plants at the bottom are not going to get sunlight and are going to start to die because, unfortunately for them, they require to do photosynthesis. So our light levels are going to decrease. The number of bacteria are going to increase because what they're going to do is they're going to start feeding off all the dead organisms. So, for example, all the dead plants and dying organisms in there, they're going to start to decay. The number of bacteria that eat the decaying material is going to start to increase. So we're seeing here there's a pattern, so we can't have that one, we can't have that one, we can't have that one. So that rules out A. Okay, that means that the only answer available so far is D. Let's just go through and double check. Oxygen levels are definitely going to decrease because the bacteria are going to start to consume more oxygen, which means that the other species that live within the loch are going to have less oxygen availability, and they're either going to go to another area, but if they can't, they might also find themselves dying as well. So question number 24, answer is D. And last question for paper 1 is the following 3 processes occur during speciation. We've got mutation, natural selection, and isolation. And the order in which they occur is what? So A, B, C, D. Pause the video, see how you get on. Okay, so we want to think about this one. So we've got mutation, natural selection, isolation, and we've got A, B, C, and D. So would it start with isolation? Yes, it actually would. So you imagine you've got a bunch of organisms here, and suddenly a mountain appears. Okay, so this 2 are going to go live over here, and this 2 are going to go live over here. And so the first step is isolation. Okay, the next step is going to be natural selection. So we've got 2. So we can already see our answer is going to be A here. But that natural selection, so maybe on this side it's really hot, and on this side it's freezing cold. Now this side is going to start to adapt to it. So they're going to start to mutate towards that environment over a long period of time. And so we're going to see that mutation happen there. And eventually when they come back together, you would see that they are different species. But again, this happens over hundreds of thousands, if not millions of years. It takes a very, very long time. It's a very slow process. And that's it. That brings us to the end of this paper. So please remember to give this video a thumbs up, give it a like, give me a follow, and if you've got any questions, pop them in the comments below and I will try to get back to you as best I can. Thank you for watching.